propose, before we close the session, since we have some time here, the session is supposed to finish at 9:30, to have a discussion on why so little theory is being done in this field. Any ideas? Yesterday there was somebody, I think in this room, from Novartis, clearly in need of optimizing the interactions of peptides with proteins. I asked him, do you do simulations? No, we don't do it. Our big pharma has done a lot of simulations in the past. 15 years ago, Bristol Myers Squibb and other companies were spending lots of money to make computers. And also, there was, I remember the name of a guy called Terry Stauch, and he did lots of calculations. I guess the results were not very encouraging at that time because it was too early, probably. Now, the simulation has done tremendous progress. Computers are much larger, and the results are much more reliable. Probably was a question of too early at that time. I'd like to ask the audience if you have any opinions on this, or if my impression that now is the time to do simulations may be wrong. Judging from the number of people in this room, actually may be, but with Dr. Venter we have discussed this before.
I mean, if you look at docking, and I think, in the early 90s, the approach was just massively oversold, right? I mean, it was, sold under the premise that it would solve the small molecule drug discovery problem, right? I think even today, 25 years later, we are clear that even if you look at this one small particular aspect, right? Key things that are relevant in receptor ligand binding, namely the conformational flexibility of the receptor, are not understood, right? The scoring functions were bad in 1990, and the scoring functions are bad now, right? If you ask somebody to give you a quantitative calculation of delta G, right, of a small molecule binding to a somewhat flexible complex receptor, then the answer is nobody can do it. Right? I mean, even D.E. Shaw, who put down 300 million euros to develop his supercomputer exactly for this purpose, still cannot do it. Right? And at the same time, the experimental techniques have gotten significantly better, and they've shifted in direction. Right? I mean, I think all of this CRISPR-Cas9, high throughput screening, human genome project, right? All pushed everything much more into the direction of genetics as opposed to chemistry, right? And on a very broad scale, I think what we can simulate relatively well is chemistry, and what we can simulate really, really bad is genetics.
Yes, I can report here that there is also my colleague Alessandro Laio at CISA. He has done small molecule docking in two polymers, and then has experimentally frozen the polymer around it and released the small molecule, and then using this cavity to recognize the small molecule. And the success has been great. They can go down to picomolar interactions in this way. And I think small molecule, though, peptide not yet. Peptide is our goal to do it. And we have a long way to go still, but we hope to get nanobodies in the small nanomolar range. We've not done it yet, but pure design. You can develop them, of course, with cameras and so, but not with design on silicon. But the small molecule has been a great success. Okay. If not, I'd like to close the session and thank you, the audience, for being here. Next time, we have a room full of people and room full of results.